Hello everyone, myself Lakshmi Das, volunteer of Applied Forensic Research Science. Here I am introducing a topic, Introduction to Forensic Biology and Serology. Moving on to the content section. First, Introduction, History, Branches, Collection and Preservation of Biological Evidence, Packaging of Evidence, Case Study and References. Introduction. As we know, forensic science is the application of scientific method along with the justice of law. It has more than 15 branches. Here we introducing forensic biology. It is the examination of biological specimens which has been recovered from crime scene. The forensic serology is a sub-branch of forensic biology. It is the examination of biological fluid. It has been become important after the discovery of DNA fingerprinting or DNA profiling. And the DNA fingerprint or DNA profiling has been discovered by Alex Jeffrey, who is known as the father of DNA fingerprinting. The history of forensic biology depends on two scientists, those who are Carl Landster and Sir Alex Jeffrey. Sir Carl Landster, who discovered ABO blood type in 1901. Those ABO blood typing or ABO blood grouping are A blood group, B blood group, O blood group, and AB blood group. And the second scientist was Sir Alex Jeffrey, who discovered DNA fingerprinting or DNA profiling in 1985. The father of forensic biology is Carl Landster, and in 1915, Lattice invented first antibody test for the blood group. 1920 to 1950s, the discovery and application of new blood groups and serum protein has been happened. The new blood group was small, another one is ER, that is a rare blood group, and it was discovered by NHS Blood and Transplant Institution in Bristol. Then in 1984, electrophoric variants of red blood cell enzyme were discovered and its use has been started. In 1991, first polymorphic human short tandem repeat have been identified. The short tandem repeat has been discovered by Kohlenberg scientist. And in 1992, the first commercial forensic short tandem repeat profiling kit was developed. And in 1992, again the first YSTR, that means the short tandem repeat, has been found on the Y chromosomes mainly in male individuals. It was described and used in a rape accusatory. Next, in 1992, first application of mitochondrial DNA in casework. The mitochondrial DNA was discovered by Margaret Nash and Selvar Nash in 1963. It was found in the eukaryotic cells. The mitochondrial DNA has been transferred from mother to children and is also known as a EVE gene. Next, in 1992, the National Research Council report on DNA technology in forensic science was published. In 1995, National DNA Database has been established in United Kingdom, and in 1997, DNA profiling of touched object and single cell has been demonstrated. Next, history of forensic serology. The father of forensic serology is Sir Arthur Moran from 1904 to 1994. He found a serum with antibody to the E factor in 1945, and his subsequent development of the antiglobulin test, which confirmed the compatibility of blood in transfusion. The antiglobulin test, also known as Combs test, which is used for the presence of antibodies against circulating RBC. And in 1863, German scientist Christian Schonbein discovered the oxidation of hydrogen peroxide when exposed to hemoglobin. It was the first presumptive test for blood, or also known as Castle-Mayer test or phenolphthalein test. The Castle-Mayer test was discovered by Joseph Hoying Castle in 1901 and modified by Erich Mayer in 1903. That's why the test is known as Castle-Mayer test. It is used to detect the conformation of a blood. Due to the presence of hemoglobin, that stain turned from colorless to pink color. It detects the conformation of blood. Next, in 1900, Carl Landster, who discovered human blood group for the first time, he awarded Nobel Prize for this in 1930, and it's also known as the Serology ABO blood grouping. In 1971, Brian Culliford published the examination and type of blood stain in crime laboratory, establishing protocol and standard method for typing of protein and enzyme makers. And 1910, Serology department was established in Calcutta. After independence, the department name was renamed as Office of Serologist and a Chemical Examiner to the Government of India. In 1957, the first forensic science laboratory was established at Calcutta, including 4 divisions, which are forensic physics, forensic chemistry, forensic biology, and forensic ballistic as a central forensic science laboratory. Next, in 1965, the second Central Forensic Science Laboratory was established at Hyderabad with 3 divisions. They are Forensic Physics, Forensic Chemistry, and Forensic Biology. Next, the branches of forensic biology. They are forensic serology, forensic DNA profiling, forensic botany, microbial forensic, forensic taphonomy, forensic craniometry, forensic odontology, forensic anthropology, and forensic pathology. Those sub-branches of forensic biologies are of forensic entomology, forensic medicine, forensic criminology, wildlife forensic, and DNA forensic. Cases which were received in forensic serology division in the laboratory are of blood typing case, characterization of unknown stain, blood spatter analysis for crime reconstruction, paternity testing, semen identification in sexual assault cases, DNA typing for identification of victim and suspect. The evidence which are received in forensic serology division are of blood, saliva, semen, sloughed skin cells, hair, urine, and fecal material. The cases where those biological evidence has been recovered from crime scene are of homicide cases, assault, suicide, rape, sexual assault cases, burglary, hit and run cases, motor vehicle accident, criminal paternity, mass disaster. These are. Collection and preservation of biological evidence. First one, blood. Colored photograph of blood stain from every direction should be taken because those photographs help in crime scene reconstruction. And always remember that each sample of blood should be packed in different packet after air dry. If wet blood stains are there, it should be dried before packing for better preservation of the sample. If blood stains are present on immovable object like a concrete substance or any other hard object, first take colored photograph and then collect the sample with the help of a sharp object like a knife or scalpel and preserve those sample in a glass bottle, polythene or a paper bag. Then, if blood is in the form of a liquid, use a dropper to collect those blood and preserve it in a glass or plastic bottle. After adding anticoagulant to prevent the coagulation of blood, we should add anticoagulant to the vial. The examples of anticoagulant are of fluoride salt, heparin, sodium citrate, etc. Then those packed samples should be stored in a cool and dark places, and those articles sent to the forensic laboratory for further examination. Doctors can collect or preserve blood on the request of investigator, and during postmortem time, doctor can preserve those blood for the later investigation also. If there is a more than 2 more suspect and those suspect use more than 2 weapons, collect those all weapons from the crime scene and collect in a different packet individually. Next, semen. The collection of spermatozoa has much precautions because semen convert into brittle after drying. So if stains are found on a cloth or any other object, if it is not cared properly, there is a chance of breakdown of spermatozoa into pieces. Then preservation of semen performed after dryness of a stain. If stain is in a wet condition, there is a chance of growth of bacteria will start and may chance of decomposition of a protein in semen. So if the stain is in a wet condition, keep it for drying and then preserve the sample. It is always remember to preserve the sample in a air bag and not in airtight bag or plastic bag. If the seminal stain is found in immovable object, then preserve those articles by using a sharp object like a knife or scalpel, and before collecting that sample, take a photograph and collect the sample with a sharp object like a scratching method and pack it in a clean glass bottle and send it to forensic science laboratory. If any infection in testis due to some bacteria also secrete liquid like a seminal stain, which creates some doubt regarding if this is a seminal stain or any other stain. So collect those stain also. Next, stain may be present on pubic hair also. So it should be preserved in a bag or any clean plastic tube. Next, never touch the sample with a bare hand because there is a chance of contaminate the specimen. If stain found on a body part, do swab method. By dip that cotton swab in a saline water, then apply that swab on the body and do the swabbing method. Keep it that swab for a dry and preserved in a air bag. Fresh seminal stain, there is a presence of live spermatozoa. So keep that sample safe and report it to the forensic science laboratory. Next biological sample, saliva. Saliva is an important sample in crime investigation because it's helpful for DNA profiling or grouping test, which can be performed by saliva due to the presence of its antigen. Blood group can be detected by saliva because saliva may be contaminated by lipstick or blood. Through that, we can detect the blood group. If the sample is dried, then wet the sample with the help of a normal water and do the swabbing method with a cotton swab and preserve that swab in a tube or bag. Then, if chewing gum, tobacco, spite found in crime scene, preserve it in an airtight bag. Next, urine. If urine present at a crime scene, then preserve the sample carefully. Some liquid create a doubt that may be a urine or not, so that liquid should also be collected in a plastic or glass vial. And if urine present in a cloth, then dry the cloth under shed means not dry the cloth in a outside, no need too much sunlight for the drying. So keep that sample to dry under a shed and preserve as soon as possible and send it to the forensic science laboratory. Fecal matter. The fecal matter is most commonly found in the case of a sexual assault cases. So, if there is presence of fecal matter is there, preserve the sample in a plastic bottle. If it is difficult to collect that sample, keep it for drying, then collect the sample. Then vomit. In a crime scene, there is a presence of vomit. It is must to do the preservation of vomit. Then if the sample is mixed with the soil, then preserve those soil with the vomit sample should be preserved with the help of a spatula and collect those sample in a plastic bottle with the suitable things. Before collecting those sample, wear gloves. Next, hair and fiber. In a crime scene, maybe hair or fiber should be present. And these are very minute sample, so need more precautions while handling. If something is present in crime scene like a hair or fiber, it should also be collected for the further examination. In a crime scene, fibers classification are of more types: vegetable, animal, synthetic, artificial fiber, anything. Those all fibers are present in crime scene. Collect those fiber in a different packet and labeled with a name and source. And collect those hair and fiber sample with the help of a forceps.